My name is Svensson. I have about 20 years of experience in pharmaceutical industry, having worked at companies like Sanofi Aventis, AstraZeneca, and now RISE. I have also long-term collaboration with KTH in Stockholm, and my expertise lies in computational chemistry, robotized screening, material science, structural informatics, and the utilization of synchrotron techniques. So my talk today, I'm intending to provide you a condensed overview of our efforts within computational pharmaceutics and robotized screening in pharmaceutical development. Short outline. First, I will provide a concise overview of the computational pharmaceutics landscape. Following that, I will highlight practical examples in some key areas that showcase the potential of computational pharmaceutics. And next, I will present some of our efforts in robotized screening aimed at speeding up the discovery of novel materials. And finally, if time allows, I will share some personal insights in the area. I will focus on the product development phase and traditional small molecule APIs. I will not cover the drug discovery phase, and ADME predictions are not covered in this presentation as well. Here we have a basic overview of the computational pharmaceutics operational landscape. It covers everything from liquid crystal to bulk systems, where the solid state system dominates. If we look into the crystalline system, there is a vast amount of computational supports with many areas, including a range of in silico screening methods. For amorphous system, there also exists numerous amount of computational support, and here also you can get insight to kinetic properties. For both particle and bulk system, there are also numerous areas where in silico support is available, however, maybe somewhat lesser extent. But overall, there is robust computational support accessible. And I have selected some examples from these areas to illustrate the computational pharmaceutics in practice. But before going into practical examples, I would like just to have a brief overview of the computational support available, which utilizes various levels of theory, ranging from quantum mechanics, molecular mechanics, as well as coarse-grained models, along with some statistical and mathematical models. And these methods covers a very wide range, both in time and length scale, and as a result, also a wide range of properties, as can be seen on the right-hand side of the slide. And instead of categorizing computational pharmaceutics by system investigated, as in the previous slide, we can also divide it into a more general way into three application areas. And this covers property, formulation and process optimization, analysis and interpretation of experimental results, supporting, for example, spectroscopy, powder diffraction, or NMR, and in general, of course, problem solving. Let's shift our focus to practical examples where computational pharmaceutics has been applied. This first example deals with crystal growth in connection with absolute configuration determination of an oncology substance, vakinol, which is active against a very aggressive type of brain tumor, glioblastoma. In this collaboration project with Karolinska Institute, we were targeted with the determination of the absolute configuration of the active substance vakinol, since it was unknown. There was, however, a small amount of diastereomere substance available, making the crystal necessary for the X-ray determination of the absolute configuration. Subsequently, a critical question emerged. Which solvent should we be using for the crystal growth experiments? And of course, with 50 milligrams of substance, you can't waste, and you have to be trying to target the right solvent at an early stage. And here you can see how the crystal looks initially, and these are very small and clustered and not appropriate for absolute configuration determination. In order to facilitate and support the experimentation, molecular mechanics-based solvent screen of the API was conducted. And here we can see the results for such a prediction. On the x-axis, we have the interaction energy, and on the y-axis, we have the so-called packing index. Each point corresponds to a solvent examined. The highest ranked solvent for the API was used in the initial crystallization experiments. And here we can see the results, and on the right-hand side, we see the result of the crystal growth, and this single crystal is mounted on nylon loop. And in principle, at least 10 times large crystals were obtained. Furthermore, we determined the crystal structure, and here you can also see that actually the solvent was included into the lattice, meaning that we got a solvate of the API. And of course, we were able also to determine the absolute configuration of the substance. Next example is connected to form selection and co-crystals, which is an alternative to salt-based APIs, and where a neutral co-former is added to the API to form the co-crystal. The question is, how can we find appropriate co-formers? Since this process can be very time-consuming, computational support would be extremely helpful to guide the discovery of interesting co-formers. By utilization of a molecular-based method, we could screen a library containing about 1,200 co-formers against the API, and in this case, it's theophylline. Here we see the outcome from the screening, and the highest ranked candidates marked with a circle in the upper left corner were investigated, and here each point corresponds to a predictive co-crystal system. And here, in the same way as before, the x-axis is the interaction energy and the y-axis is the packing index. And the four highest ranked candidates was experimentally evaluated, and 3 resulted in co-crystal formation. An example of a co-crystal formed with salicylamide can be seen on the right-hand side. It is noteworthy that the library also contains co-formers that is not suitable for pharmaceutical development. However, the inclusion allows for comprehensive understanding of what can trigger co-crystal formation in different systems. And an example of such a library candidate in this case is cyanophenol. And in this case, we were not actually able to get a co-crystal with this co-former. Next practical example deals with intrinsic solubility and solubility improvement. And if we look at the solubility from a solid state point of view, there are 2 factors affecting the solubility. The entropy that have a positive effect of the solvatization, while the interaction energy has a negative effect. So what can we do to reduce the interaction energy to increase the solubility? The solution that we introduce is a methodology where we perturb the packing or key interaction in the crystalline state to reduce the interaction energy. And here is an example of a perturbation prediction where different perturbation have been applied. And here the overall aim is actually to reduce the difference between the amorphous and crystalline solubility. And in the box on the right-hand side, we can see different perturbations. And the result, as I said before, is shown in the plot. One example of a perturbation could be, for example, exchange of hydrogen to fluorine. And with this approach, it's really aimed for so-called brick dust compounds, which has a high melting point. And in principle, you can achieve up to 20 and even more times improvement of the solubility with this method. I will just show you a very basic example here of the perturbation approach on the benzodiazepine series, where we could show that it's possible to predict the effect of different perturbations of a larger series. The perturbations are marked with circles, and going from compound 1 to 4, there is a solubility improvement of about a factor of 10. This has been published some time ago, and if somebody's interested in the references, please don't hesitate to contact me. The perturbation methodology can be used also in the reverse to promote crystallinity, and in this case, we want to increase the interaction energy. Well, I don't have any examples of this, but it's the same principle, but we really work to find more crystalline material instead. Let's go to another example, which is dealing with surface inhibition by impurities. And in this example, there was seen a deviation in dissolution rate between 3 different batches. But we couldn't understand why. The batches was of high and similar purity, but in one of the batches, there was actually a very small amount of impurity that had a very similar molecular structure compared to the API. With MD calculation, it was actually possible to discover that this extremely low concentration impurity have the perfect fit in the surface of the API. And this would, of course, have an impact of the dissolution rate. And here in the figure, you can see the impurity fits very well into the surface of the API and also interacts very well. And we also made further studies of this case, and we could actually see that the impurity formed a solid solution, meaning that the impurity actually incorporates into the crystal lattice. And this is, of course, a very extreme situation and can create a very complicated situation. Finally, I will show 3 more general shorter examples of utilization of computational pharmaceutics. The first one is connected to the morphology and size of the API crystals that have an impact on many critical parameters, especially particle and bulk properties. Therefore, insight in how to control and manipulate the morphology can be very useful. And here I just show you a very basic prediction of the morphology, and on the right-hand side, you can see that it's at least a reasonable coherence with the actual crystal formed. The second is an example related to mechanical properties and how it is possible to track when and in what direction the mechanical properties can have issues, and where the API structure can collapse under application of external pressure. This capability can be very helpful in risk assessment and process development. And here is mentioned the Young modulus, often denoted as E. It is a measure of the stiffness and elasticity of the API and material investigated. And finally, a brief example of how varying mixtures of API and excipient can influence stability and solubility. And here we see the MD calculation of the API and excipient in liquid state and without the solvent included on the right-hand side. And in this case, by doing analysis of the hydrogen bonds and the conformation and a conformation analysis, it was seen very useful in understanding the system's stability and also the effect it had on the solubility. Having discussed computational pharmaceutics, I would like now to turn our attention to robotized screening. I will try to give you a short overview of our efforts in this area. In this slide, you see one of the systems that we have been working with. So why should we use robotized screening? Currently it takes an alarming long time to find novel and suitable, sustainable materials, and consequently there is real urgent need to development and progress in this area. And to meet these challenges, we have for many years been engaged in screening efforts and concurrently pursuing an initiative to develop a novel robotized screening platform to accelerate the discovery of novel and sustainable materials in different key areas. And then by employing robotized screening, we really can accelerate the identification of islands within compositional and chemical space that exhibit unique and desired material properties. And the areas we have been applying this to is different type of screening, salt screening, co-crystal screening, also reaction crystallization, crystallization scale-up, but also quite a lot in recent years in material screening. And here on the right-hand side, you can see a previous platform that we have been working with in action. However, this system has several significant disadvantages. It is very expensive, it has high maintenance, and it's actually very space demanding. And to develop this platform further is very challenging, and if we would like to implement AI or machine learning, it would be very difficult. And furthermore, it also generates a significant amount of waste. However, depending on the really explosive development in the field of robotics and AI and machine learning, it's now possible to design much more intelligent and compact, as well as versatile screening platforms that can replace these larger systems with smaller modular systems with AI capabilities. This type of modular system can be adapted for more specific tasks and at a relatively low cost, opening up for exciting opportunities for faster detection of new compounds and in various domains. This type of modular system has also other important advantages, such as much less use of material. Of course, the implementation of machine learning and AI is much more easy, and we could therefore also go forward to create an autonomous platform. And this new system also has very high repeatability and throughput. And currently, we are looking into developing screening platforms connected to precision medicine, utilizing 3D printing, and also traditional formulation of screening. And when it comes to 3D printing, we are particularly interested in implementing automated quality control, which includes imaging and spectroscopy. Efforts to develop the overall new control system for the platform is currently ongoing. And this slide is just to illustrate how schematically, how module-based screening platform can look like. And reaching for a new system, we have, of course, been working with different types of prototypes systems, and this you can see on the left and right-hand side. Finally, I would like to mention some personal insights to what I believe can be done to further accelerate the development and unlock the full potential within the title area, and potentially being able to advance forwards an autonomous laboratory. And I believe that the integration of computational modeling and experimental workflows can streamline the process and guide the experimental design, and thereby reducing the number of trial and error experiments, and thus accelerating the identification of promising candidate materials and substances. We need more tailor-made models for specific systems to speed up the calculations. Many approaches are currently quite generic, which also reduce the precision. Moreover, we also need to facilitate access to critical data information via knowledge bases, and additionally, expediting the validation of new models is imperative to access success here, and we must generate a greater volume of high quality data, which is key to really develop machine learning models. And finally, we need to implement more high throughput characterization techniques, for example, high throughput microscopy and spectroscopy, as well as X-ray diffraction, to rapidly analyze the properties of materials and screen larger sample libraries. And I would like to have one takeaway in this talk, and in my belief, it's the importance of being able to integrate in silico screening and robotized screening with AI and machine learning to accelerate pharmaceutical development and advance towards the possible realization of a more autonomous laboratory.
Which of the areas you presented do you believe has the greatest potential in the long run, or in the short run?
I think currently leveraging AI and machine learning algorithms for data analysis, predictive modeling, and decision making for screening processes can really significantly enhance the efficacy and accuracy in the area. So this is happening so much in this area. So I think this is really explosive. And then, of course, advances in robotics and automation technologies will enable very high throughput. So there's a lot of things happening in this area.